Hello, welcome to ninth episode of neuroanatomy series. Today we are going to focus on basics of peripheral nervous system. So let's see what is nerve, how it is composed of, what is components of nerve. As I said earlier that peripheral nervous system is the extension of central nervous system. You can say the central nervous system sends the feet in all over the body like octopus to make a connection with the rest of the body to send the signals to the body and to receive the signals from the body. So the same neurons that make up the brain extend as nerves and make up nerves. So the neuron is the nerve cell, as the neuron is the brain cell or spinal cord cell. Neurons arise from brain and spinal cord and run as nerves and end up there, and from there other neurons start. So all the parts of the neuron are included in the nerve. But because the axon is the longest part of neuron, so most of the nerve is composed of parallelly running bundles of axons. And that is why the axon is called nerve fiber. This is nerve fiber. These nerve fibers bundle up to make a fascicle, and this fascicle is enclosed by perineurium. And the connective tissue layer that surrounds the individual nerve fiber is called endoneurium. Nerves also have blood supply and neuroglia in it, and all these contents are surrounded by a layer that is called epineurium, the outermost layer of dense irregular connective tissue that surrounds peripheral nerve. It has the cushioning effect and gives the tensile strength to the nerve. Keep in mind that as the bundle of nerve fibers are called nerves in peripheral nervous system, the bundle of nerve fibers in the central nervous system are called tracts. These tracts are pathways that carry information up and down. We will be talking about them in detail when we will be talking about the tracts. Now coming back to nerves. This is the neuron whose cell body is in the CNS, and this is the neuron whose cell body is in the PNS. Neuronal cell bodies in the CNS are called nuclei, while the neuronal cell bodies in the PNS are called ganglia. Means the neuronal cell bodies in nerves are called ganglia. There are 12 cranial nerves and 31 spinal nerves. Cranial nerves start in the brain and distribute mostly in the head and neck area, except 10th cranial nerve that goes down to the thorax and abdomen. Some cranial nerves are sensory, some are motor, and some are mixed. On the other hand, the spinal nerves start from the spinal cord and distribute to all over the body. And all the spinal nerves are mixed, means they are made up of sensory as well as motor neurons. These sensory and motor neurons communicate via an interneuron that is also called associated neuron. Most of the interneurons are found in CNS, while some are found in autonomic ganglia. As I said, like CNS, peripheral nervous system also have neuroglial cells. These are Schwann cells, satellite cells, enteric glial cells, and olfactory ensheathing cells. Schwann cells has same function as oligodendrocytes in CNS. They are responsible to make myelin around the axon, but they also play a vital role in development, maintenance, function, and regeneration of peripheral nervous system. Myelin is a fatty substance that is non-conductive. Let me show you how myelin increases the velocity of impulse. I told you in episode 6 of neuroanatomy series that was regarding action potential that action potential is propagated through nerve through voltage gated ion channels. These voltage gated ion channels are distributed throughout the nerve membrane in non-myelinated axon, but these are only concentrated in the node of Ranvier in the myelinated axon. Nodes of Ranvier are gaps formed between myelin sheath where axons are left uncovered by oligodendrocytes in CNS and by Schwann cells in PNS. And this is done on purpose because if they were not left uncovered, the propagation of action potential would have been impossible, as the propagation of action potential needs voltage-gated ion channels throughout the membrane. When the action potential is present in non-myelinated axon, the ions are lost because the channels are present throughout the membrane. But the escape of ions is pretty much impossible in the myelinated axon. As the channels are just concentrated on the nodes of Ranvier, the propagation of action potential just happens on nodes of Ranvier. And that's how the waste of current is less and the speed of current is more. But not all the nerve fibers are myelinated. In the fibers we need slow conduction of the current, those fibers are unmyelinated. As there are myelinating Schwann cells, there are non-myelinating Schwann cells. They don't have the function of myelination. Instead, they are responsible for maintenance of axon and neuromuscular junction, peripheral nerve regeneration, and remodeling of neuromuscular junction. Other neuroglial cells are satellite cells. They cover the surface of neuronal cell bodies in ganglia of peripheral nervous system. However, main function of satellite cells is still not known, but they are thought to control microenvironment of sympathetic ganglia. Coming to enteric glial cells, they are associated with the cell bodies and processes of enteric neurons, means neurons that are associated with the digestive system. They exert essential roles in supporting the survival and function of enteric neurons. And lastly, olfactory ensheathing cells. They are responsible for regrowth of olfactory axons. They successfully regenerate the olfactory axon throughout the life. These were some basics of peripheral nervous system and nerves. The journey of neuroanatomy is continued. See you in next video. Till then, Khuda Hafiz.